Hello, students. Good morning, everybody. Today, I'm going to talk about bryophytes. A general introduction we will talk. I'm Dr. Mandeep Kaur from Guru Nanak College, Sri Muktsar Sahib. Bryophytes are basically an informal group consisting of three divisions of non-vascular land plants, that is the liverbirds, hornbirds, and mosses. They are characteristically limited in size and prefer moist habitats, although they can survive in drier environments also. They comprise of about 20,000 plant species. They have enclosed reproductive structures, that is gametangia and sporangia, but they do not produce flowers or seeds, and they reproduce via the spores. Regardless of their status, the name is convenient, and that is why bryophytes is still used. Now, term bryophyta, this was given by, this means from Greek, it means tree, moss, or oyster green, and phyton means plant. Bryophyta term was given by Brown in 1864, and later G.M. Smith, he placed this group between algae and pteridophyta. Bryophytes, they exist in a wide variety of habitats. Here you can see them growing in a forest. They are growing on the forest floor as well as on the tree trunks also. They can be found growing in a range of temperatures, that is the cold Arctics and hot deserts, elevations sea level to alpine regions, and moisture dry to wet rainforests also. They can grow where vascularized plants cannot grow because they do not depend on roots for uptake of nutrients and water from the soil. They have rhizoids, and they can survive on rocks and bare soil as well. Here you can see in this picture they are growing very well in rocks, on the rock crevices. This is on the tree trunks. Now, bryophytes, they have distinct features like their life cycle is dominated by multicellular gametophytic stage. This is the gametophytic stage of Rixia. Here we have in the case of Mercantia. This is the Anthoceros thallus, you can see. This is the sphagnum. Here we have Funaria. Right, Funaria is leafy, and this is the sporophyte region. We will talk about it later on. They have rhizoids. They can be smooth rhizoids, smooth-walled, or pegged or tuberculate. Then Mercantia, etc. They have, what do you call it, scales also. The sporophytes, they are unbranched, and generally they are dependent on the gametophytic stage. They do not have true vascular tissue containing lignin. Right? So one by one we will talk about all the three groups. Liverbirds, they are called Mercantiophyta. Here we have Mercantia, and Gamicups are visible over here. They have about 9,000 species, small non-vascular spore-producing plants. They are distributed worldwide, and most commonly they are in the tropics. They have a thallus or thalloid body, and it resembles the liver lobes, and that is why it is called a liverbird. Examples are Rixia and Mercantia. Here we can see the thallus of Mercantia. On the undersurface, that is the ventral surface, they have rhizoids as well as the scales. Scales can be of two types. Then the Gemicups are there for asexual reproduction. They contain gamete, which are dispersed later on. Then the female gametophyte and the male gametophyte, they can be on the same plant, or they can be on separate plants as well. They are present on, this is called the anthridiophore, where the anthridia are present. Here we have the archegoniophore, where the archegonia are present on the underside. Afterwards, from the gametophyte, the sporophyte will be formed. We will talk about the life cycle later on. Here I am showing the spores and elaters of Mercantia are visible. The orange stained are the spores, and these elongated, spirally coiled, these are the elaters. These are useful for the dispersal of the spores due to their hygroscopic movements. Now here we have the Spores and elaters of Tylidium pulcherinum. This is also a member of the Marcantiae family. Next, we come to the hornworts. Hornworts are also a group of bryophytes which belong to the division Anthocerophyta, and they are called hornworts because of the elongated horn-like structure of the sporophyte. Now here these are the flat thalloid surface, that is the gametophytic structure. Over here also you can see, and these are the hornworts, or you can call the sporophyte. Sporophytes, they have intercalary meristem, and so that is why they grow quite long. And the main plant body over here, again, it is a thalloid gametophytic plant. Examples are Anthoceros and Dendroceros. So here you can see this is the flattened surface of the gametophyte, and rhizoids are present on the undersurface of the gametophyte. Then these are the sporophytic portion, sporophyte, and spores are produced inside, and they have a columella also going throughout the length of the sporophyte, and they have intercalary meristem over here. Here we will see these are the spores being released through the upper part portion of the sporophyte after the longitudinal slits and spores, as well as they have pseudo-elaters. So these are the pseudo-elaters of Anthoceros. Next we have the mosses. Now mosses, they are also called mussae, and they are also a small division of the non-vascular flowerless plants, Bryophyta. Mosses, they form dense green clumps or mats, often in dense and shady locations. Individual plants are generally composed of, they have foliage, foliose structure. They are not thalloid, and they have simple leaves, generally one cell thick only, and attached to the stem. In the case of Funaria, you have the main stem which acts as the male branch, and the side branch acts as the female branch, and from the female branch you get the sporophyte production later on. So here we have this is the examples are Funaria and Polytrichum. Here you have the rhizoids on the lower surface. This is the main axis, and these are the leaves. And this portion is the gametophyte, and the upper portion is seta and capsule. Foot is embedded inside the gametophyte. Now we will see the peristome teeth of Funaria. This is a enlarged view and close up of the peristome teeth, not the peristome teeth. Here we have the basically the capsule. This is the capsule, and upper portion is called the operculum. And inside the operculum, you have the peristome teeth, right? So this is a mature capsule, and it changes its color from green to red or brown. Here the operculum has fallen off, and these are the peristome teeth. In the case of Funaria, you have two rows of 16 peristome teeth in each, so total 32 peristome teeth are present in Funaria, and due to their hygroscopic movements, the spores are released. Here is another picture of the peristome teeth of Naria, and this is in the case of Bryum. Now we will have a quick look at the life cycle of liverworts. We will do these in detailed form in other lectures, but I just wanted to give you a short introduction. So this is the male gametophytic thallus, and this is the female gametophytic thallus. They have the archegoniophore, and here they have the anthridiophore. So in this case, after the inversion of the uh, this archegoniophore, you have the inverted position of the archegonia, and then you have the neck canal cells and cover cells and the venter and egg inside the archegonia. In the case of Anthridium, these have this is a club-shaped anthridium, and it contains various anthridiozoids. After these are already haploid plants, so there is no need for meiosis formation. So haploid antherozoids and haploid egg, and later on they undergo fusion, and a zygote is formed, which will be here inside the archegoniophore itself. So over here only it will develop, and there will be a short foot and a seta, and you will have a capsule over here which will be covered by the tails for protection. And inside, after meiosis, the spore mother cells will form the spore, tetrad, and spores. They will have exine, etc. And you will have the elaters also along with them. We have seen them already. And after the dehiscence of the sporophyte, spores will start germinating, and you will get the formation of thallus again. So this is the life cycle of liverworts in general and Markensia to be specific. Here we have the structure of the archegoniophores and enthridiophores. These are the real pictures, and then their fusion and the sporophyte formation starts over here. This is the sporophyte hanging down from the archegoniophore. In the case of Funaria, we have these are the rhizoids and the male and female branch of the gametophyte. And then afterwards, what happens over here is inside the involucre of the leaves, you have the anthridium, and inside the involucre of the leaves and pericyclical leaves, etc., you have the archegonia. And with the help of the water film, the sperm will move and reach the archegonia. So water is very essential for the reproduction and life cycle completion in all the bryophytes. And then after fertilization, zygote will form over here, and sporophyte development will start. Zygote will be the first cell of the sporophyte, and you will get the sporophyte formation, and eventually you have a foot embedded inside and a long stalk which is called seta, and the developing capsule. Later on, initially it is erect like this. When it gets ripened or mature, it will droop like this, and later on the operculum will fall off like this. Inside this you will have spore sac, and where the spore mother cells will undergo meiosis and spores will be formed. There is a columella in the center, and the seta and the outer layer of the capsule is green, and so it is also photosynthesizing, although it is dependent on the gametophyte in the initial stages. So when the spores, they are formed, and after the hygroscopic movement of the peristome teeth, etc., and shaking, the spores are dispersed, and then germination starts, protonema formation starts, and like this you get the, after protonema, you get the proper gametophyte stages, the branched stages. So this is in the case of Funaria. Polytrichum also has a similar life cycle. Next, we come to the Anthoceros. Now, this is the thallus of Anthoceros. There are rhizoids below. They do not have scales, and they have embedded anthridia and archegonium. So, inside the thallus, you have the archegonium over here, and covered anthridium, you have over here biflagellate sperm. This will also swim in a film of water and reach the egg, and ultimately the development of the sporophyte will start. This sporophyte is long. This has a foot over here, and after the foot you have a meristematic region which is called intercalary meristem. Due to this, the sporophyte grows quite long compared to the other bryophytes, and even compared to its own size of the gametophyte. And you have a central columella and spore sac on both sides of the columella, and spores will be formed after the meiotic divisions, and these will be released just like I had shown you in the earlier photograph. I will just show you again. Like here, the longitudinal slit will be there, and the spores will be released along with the pseudoelaters will help in them, right? So now there will be a quick recap of the structure of liverworts and mosses and hornworts. They are gametophytes. Structure of liverworts and hornworts, this is thalloid. However, there are some liverworts like jungermanials, etc., which are foliose. So the dorsiventral or radial symmetry is there in liverworts. Radial symmetry in mosses, and hornworts have dorsiventral symmetry. Rhizoids are unicellular in the case of liverworts as well as hornworts. Then protonema is reduced in liverworts. Protonema is present in the case of mosses, but there is no protonema formation in hornworts. And gametangia are immersed in the case of hornworts, while they are superficial in both liverworts and mosses, as we have already seen in the previous slides. In the case of sporophytes, the structure is of the stomata are absent in liverworts. Mosses, they have stomata in the sporophyte. Hornworts also have it, and they are green and doing the photosynthesis also. In the case of structure, we have already done mosses and hornworts. They have large sporophytes, and they are mostly persistent in the case of mosses and hornworts. And growth is defined in both liverworts and mosses, but not in the case of hornworts because they have intercalary meristeme and it continues to grow. Capsule is simple in liverworts, and however it is differentiated and well developed in the case of mosses by operculum, peristome, teeth, etc., and theca formation, etc. In the case of hornworts, it is elongated. Then dispersion of the spores in liverworts is by elaters, by peristome teeth in mosses, and pseudoelaters in hornworts. Columella is present in both mosses and hornworts, and dehiscence of the capsule is by longitudinal or irregular in liverworts, transverse slits in the case of mosses, and longitudinal in the case of hornworts. So I hope you people have understood all the cases of the examples of liverworts, then the anthocerophyta or the hornworts and the mosses. We will talk about them in further detail in the next lectures. Thank you very much.